I am Shernan Holton from the University of Minnesota. It is the honor of a lifetime to be able to give you this talk on novel insights in the biology of steroid refractory acute graft versus host disease. By way of disclosures, I have performed consulting for Insight and Generon. I am receiving research funding from Insight, and I serve as a clinical trial adjudicator for CSL Behring. High-dose steroids remains the first-line therapy for life-threatening acute graft versus host disease. There is an obvious benefit to these. Glucocorticoids reduce activation and proliferation of effectors and reduce inflammatory cytokines, and completely resolve acute GVHD in some patients. But there are many downsides to high-dose glucocorticoids. They impair the function of regulatory cells, they impair mucosal regeneration, and some T cell subsets that could be pathogenic in GVHD, such as Th17 cells, are intrinsically resistant. So the paradigm may be shifting. Before we go further, I'd like you to stop and think about what may be happening when patients don't improve with the use of high-dose steroids. Truthfully, we don't have all of the answers. The definition of steroid refractory acute graft versus host disease is a clinical and descriptive diagnosis that is not based upon known pathophysiology. This clinical description dates back several decades and is conventionally defined as worsening or progression of symptoms after 3 days, persistent grade 3 to 4 acute GVHD after 7 days, or persistent grade 2 acute GVHD after 14 days. There are some biomarkers that can lend insight into whether someone may be likely or not likely to respond to high-dose glucocorticoids. These are helpful, but they are not diagnostic for the entity of steroid refractory acute GVHD. The Ann Arbor score, comprised of ST2 and reg3 alpha, can be helpful. We have studied amphoregulin levels in collaboration with Insight. Fecal calprotectin can be useful to monitor luminal inflammation, and a recent study showing bowel wall thickness by ultrasound may be a helpful surrogate as well. But again, none of these tests are diagnostic for the entity. The therapeutic landscape for steroid refractory acute GVHD has evolved over the decades to include more options focused on repair and regeneration. So starting in the 90s, we're focusing on broad immunosuppression with cyclosporin, ATG. Monoclonal antibodies against the IL-2 receptor and TNF-alpha were developed, for example. In the 2000s, we have additional targets for T cells, but then we start to see the entry of mesenchymal stem cells for repair and regeneration. In the 2010s, we see additional anti-T cell and anti-cytokine type of therapies, but then we also begin to see targets for trafficking, and HDAC inhibitors are being tested as well during this decade. We also see expansion then of new repair and regeneration modalities, alpha-1 antitrypsin, lithium-resistant starch, fecal microbiota transplantation, and then placental-derived stromal cells, which may be more immunosuppressive or have better repair capabilities than conventional MSCs, were starting to be tested and published. In the 2020s, we see additional anti-T cell targets and expansion again of repair and regeneration modalities, such as IL-22, and we have recently reported on the use of urinary-derived human chorionic gonadotropin and epidermal growth factor. So over the decades, largely this is focused on broad immunosuppression, but the field of steroid refractory GVHD is starting to include additional repair and regeneration modalities that I think are continuing to improve outcomes. So in truth, we don't know the full pathophysiology of steroid refractory acute GVHD. And so as I go through this talk, we'll be reviewing potential hypotheses and the data that is supporting them. So the first hypothesis that we will look at is that steroid refractory GI GVHD is just severely damaged mucosa. A couple of years ago, I published on the mucosal transcriptomal profile of steroid refractory GVHD. This was a study of 22 patients, all of whom had stage 3 to 4 lower GI GVHD, and they had paired rectosigmoid biopsies. So we had a biopsy at the diagnosis of lower GI GVHD, and then a second biopsy when they became steroid refractory. There was a median of 19 days between the first and second biopsy, and median survival of 83 days from GI GVHD diagnosis. You can see the Kaplan-Meier estimates on the right. We compared these biopsies to 10 normal controls. So the first analysis that I want to show you with that RNA-seq study is immune cell deconvolution using a tool called CyberSort. And we're going to be comparing these three groups of patients. Again, normal controls in green, GVHD onset in blue, and refractory GVHD in gray. And you can see that our arrows are pointing to T cell subsets here, CD8 and memory activated CD4 cells. There is no increase in these cells in the biopsies by this method in steroid refractory patients. Rather, what we see on the right is accumulation of M2 macrophages in steroid refractory acute graft versus host disease. Looking at the top differentially expressed genes in refractory versus onset GVHD, we see multiple genes associated with cellular stress and DNA damage. So increased expression of scARNA4, 2 metallothionein genes, and mitochondrial encoded tRNA gene all increased in steroid refractory GVHD. And then several genes that are decreased or differentially expressed and lower in steroid refractory GVHD suggest impaired tissue tolerance, such as lower expression of IDO1. Also lower expression of interleukin-1, which is a microbial carbohydrate that's involved in pattern recognition and this carbohydrate binding. That is lower in GI GVHD that is steroid refractory. To further expand upon these analyses, we're doing a new study of patients with GI GVHD with varying severities. The previous transcriptomal study was only those who had developed steroid refractory GVHD. Now we're able to look at patients who have an initial response. We call them first-line responsive. We can compare these biopsies to patients who have repeated flares of GVHD that respond to immunosuppression. We call these patients second-line responsive. And then we can compare to patients who clearly die of GI GVHD. These patients are steroid refractory and their symptoms have never improved since the beginning of immunosuppression. We call this group fatal GVHD. And so showing you some of this preliminary data here, we have 17 patients with first-line responsive, 8 patients with second-line responsive, and 4 with fatal GI GVHD. And just basically what we're starting to see is in those who have second-line responsive GVHD, they do have a higher inflammatory response and increase in genes associated with angiogenesis. They also have decreased expression of IL-20 family signaling genes in second-line responsive compared to those who just have a complete response to first-line therapy. In fatal GVHD, the principal finding is greater than 20-fold lower expression of Reg3 alpha. There are no genes that were significantly increased in expression. It was just showing to our thought that the mucosa is severely damaged and that immunosuppression alone cannot restore the mucosa. And for these patients, active regenerative measures could be what is necessary to help improve survival. What can we learn from mouse models? There are few mouse models of steroid refractory GVHD, but this one in particular did a really extensive study of T cell subsets in a steroid refractory model looking at spleen, liver, and gut T cell subsets and function in mice who were responsive versus refractory to dexamethasone. And what they saw is there's no difference in the number or function of CD4, CD8, or Treg cells in these mice. The conclusion from this mouse model was that T cells were dispensable for steroid refractory GVHD. Let's move on to hypothesis 2. Steroid refractory GVHD is caused by profound dysbiosis. This is an unsupervised clustering of microbial reads from our previous RNA-seq study. Basically what we did is we took the unmapped reads from our published study and then ran path-seq on these reads. And what we saw was that patients who had low microbial reads actually had earlier death in this cohort of patients with steroid refractory GVHD. Looking into the stool microbiota, we've identified a pattern of microbes that are associated with steroid refractory lower GI GVHD. Using linear discriminant analysis, we found that a number of bacteria, including Bacteroides, Lactobacillus, and Akkermansia, here in red are associated with refractory GVHD, whereas in blue, Doria was associated with responsive GVHD. And we found that a decline in the Doria to Akkermansia ratio preceded steroid refractory lower GI GVHD. In our previous study of urinary-derived human chorionic gonadotropin, we found that a potential surrogate for restoration of beneficial commensal microbes, such as plasma butyrate, was increasing over time in patients who responded to therapy. So in the top series of graphs here, these patients had a complete or partial response, and the bottom, these patients had no response. And we saw an increasing concentration of plasma butyrate over time in complete responders. There's some compelling evidence that Enterococcus domination is associated with life-threatening GVHD. This is a global study, really nice collaboration showing that high Enterococcus microbial contents are associated with GVHD mortality, and enterococci depend on the availability of lactose, and in this mouse model, mice fed lactose-free diet actually had improved survival to those who had a lactose-containing diet. Hypothesis 3. Steroid refractory GI GVHD is driven by steroid-resistant T cells. So there's no question that T cells cause graft versus host disease. There have been some really beautiful models recently showing that clearly the initial targets of T cells are intestinal stem cells and Paneth cells within the crypts. So of course they cause GVHD, but are they responsible for steroid refractory disease? There is some human data that shows that the accumulation of Th17 cells is associated with lack of response to high-dose steroids in lower GI GVHD. So in this box on the right, we see mucosal infiltration with Th17 cells compared to lower infiltration here on the right. And the abundance of these cells was associated with steroid refractory disease. Now recall that these cells are intrinsically glucocorticoid resistant, so potentially these could play a role in steroid refractory GVHD. There was also a recent mouse model that showed that IL-22 from TC22 cells mediates dysbiosis and is associated with fatal murine steroid refractory GVHD. So in previous slides, we showed that IL-22 has a role in repair and regeneration, but it could also promote TC22 differentiation, which could potentially drive neutrophilic infiltration and dysbiosis, at least in this mouse model. So what they saw was that administration of 1 or 4 doses of dexamethasone led to accumulation of these IL-22 positive CD4 and CD8 cells in this steroid refractory model. So this is an extremely brief whirlwind tour of some recent data looking at the pathophysiology of steroid refractory GVHD. Honestly, we are just starting to disentangle this more because there are not enough studies that are done on human tissues, and mouse models may not fully recapitulate the human experience. But what we can say so far is that there are 3 Ds of steroid refractory acute GVHD. The first is damage. There's a mucosal gene expression profile that indicates stress responses, accumulation of M2 macrophages, but not persistence of T cells based upon human mucosal biopsies of steroid refractory GVHD. Dysbiosis is clearly present. This is evidenced by a decreasing Doria to Akkermansia ratio, Enterococcus expansion, and potentially other markers of dysbiosis. Increasing plasma butyrate may indicate improvement in microbiota injury with successful therapy. And we have to be aware of different T cell subsets that may be at play here. Some studies are suggesting that Th17 and TC22 cells might be pathogenic in this condition, and they are intrinsically steroid resistant. They need further study to identify and selectively deplete if they are truly driving steroid refractory GVHD in humans. Thank you again so much for this opportunity, and I look forward to interacting with you online.